Thank you, Keith, and thank you for that very warm welcome. And thank you for coming out on a precious evening, so I hope you consider it worthwhile by the time we've been through. So to some of you, perhaps talking about human genetics, which sounds nice and hard and concrete and rigorous and scientific, and the image of God, which might sound nebulous and religious and faith-based and metaphorical, do these things really fit together? So hopefully we will be able to understand what the theological concept of the image of God means and why it matters, and also to get a better understanding of the scientific understanding of human evolution, because I'm taking the genetics aspect from the viewpoint of events in human evolution. Now you might wonder, how come a scientific pathologist working in a medical school is sticking out to talk about evolution? And hopefully the reason will become clear later, but I'll just say it now so you don't walk out straight away. My reading of scientific pathology, in particular the development of cancers, led me in a direct path into some of the extraordinary new genetic evidence for human evolution. It was the same principles. One day I was reading about how human tumors develop, and the next day I discovered the new publications coming out establishing in a brand new way the fact of our evolution. And I must also say that probably the fact of our evolution is not a surprise to people here in Britain. Even though in New Zealand we are really British, a lot of information or misinformation crosses the Pacific from America. And so we get American ideas perhaps more easily than we get British ideas from the opposite end of the world. So in New Zealand, the fact of human evolution is a much more contentious issue among many Christian people than it is amongst Christians in the United Kingdom. So I hope you nevertheless find it very interesting how the new genetics has established human evolution with such astonishing clarity and without ambiguity. So first of all, I'd just like to start off with a few comments. I'd like to work towards the idea why I think our genetics, our evolution, is a very important thing from a Christian point of view. And here we have a page of text. It's actually Greek from the Codex Sinaiticus, and it's a very early manuscript from the New Testament of the Christian faith. It's a text which is authoritative for Christians because the early church looked at the New Testament and said, this is our authoritative account of the life and work and death and resurrection of Jesus of Nazareth. Whoops, sorry, I have this quotation here which tells us about the story which came from the New Testament. The early Christians did not believe with speculations about the metaphysics of the incarnation. They began with stories about Jesus, about those whose lives got caught up in his life. We're talking about stories today, so we'll come back to that. By telling these stories, we come to see the coherence and significance of our lives as a gift. So the New Testament, it's authoritative, it tells us stories which can give our lives meaning. Now here's another text. That's the great Isaiah scroll from Qumran. And it's an example of one of the ancient texts from the Old Testament. And of course, to Christians, the Old Testament is authoritative because it was the only scripture that Jesus knew. And here's another quote which tells us that stories come from the Old Testament. Israel told a story, and it came to see itself as a people on a journey, on an adventure. It does not just happen that God's people tell stories. Story is the fundamental means of talking about and listening to God. But I'd like to suggest that there's another testament tonight, which should be authoritative in its own way for Christians. And we could call this the primal testament. It's not in the Bible, but it has a text of its own. And this is genetic text. And if you can see really close, there are only 4 letters in this text. There are A's, C's, G's, and T's. And this is actually the chapter from our genome which encodes what my students know as my favorite protein, the p53 protein, or the p53 gene. And I'd like to suggest that for Christians, this text is authoritative. We need to listen to what it says. And why is it authoritative? Because if we believe that we are created by God, if we believe that we are made in the image of God and we are created by God, then what this text tells us is binding. It's an authoritative text, even though we mightn't plonk it into our Bible. And it tells a story, another text which tells a story. The study of life, otherwise known as natural history, is storied. I want to tell you stories today from our genetic text, which help us to understand more about ourselves. We read that story in our genes, fascinating stories, which are authoritative in their own right. The Earth story, says Holmes Rolston, becomes memorable, cumulative, and transmissible in genes. So that's my basis as a Christian, and it's my basis as a scientist who is a Christian, that the genetic text, although in a different way from the New Bible texts, has its own story. And now just a comment or two. What about the image of God? What does that mean? I thought I'd talk about that at the beginning of this lecture because we need to explain it. The image of God is an Old Testament term which arose from common parlance in the ancient world. And kings set up statues. Here's one of them in the ancient world. Kings set up statues of themselves to remind people that even if the king wasn't around, here was a statue that represented the king. So wherever the king was, your local statue reminds you who's in charge. The king's in charge, so you'd better obey him. That statue represents the king. So the statue represents the king's power and rule. And when the early writers of the Bible spoke about humanity being made in the image of God, what they were intending to say in the historical context was, believe it or not, we human beings, we are images of the king. We are representatives of the king. And whether we can see the king or not, when we look at humanity, we need to remember that we represent the Creator, the King. So the image of God concept was a very important concept, establishing that humans represent God. And it falls in 3 ways. In fact, I believe, or my reading can kind of tell me that the image of God concept varies according to which of those 3 texts we're talking about. The end of the biological story leads, and if you start at the beginning of the Bible, to the appearance of humanity. Humanity is introduced as the image of God. And we could say, I'm not trying to correct Scripture, but here's just a word of my interpretation, we could call humanity in some sense a provisional image of God. And it's only mentioned 3 times in the whole Bible, the image of God concept, there at the start, but mentioned twice else. The history of Israel then leads to a brand new understanding of the concept. The writers of the New Testament say, out of Israel's history has come a new image, Jesus Christ, who is the express, the perfect image of God. Jesus Christ is everything that human beings failed to be. And then when you read the New Testament, we read that the writers of the New Testament believe that at the end of history, God would confer the perfect image of Christ onto human beings who represent God's image so very, very imperfectly. I mean, God was really put himself on a limb when he spoke of human beings and all our selfishness and greed as being his image. So there's a lot in the New Testament which speaks that one day the perfect image of Christ will be conferred upon us who bear his image. And that likeness of Christ can be given to people as a continual process, a growth in obedience to the Spirit of God, and ultimately as climax. I won't stop and read these verses now, but Christians look forward to the day when they will receive in a perfect form the very image of Christ. So that's an introduction. The image of God is an important concept. Let me just tell you one quick story before we start thinking about the genetics. About 15 years ago, the leaders of the untouchables in India came to a Christian scholar and missionary called Philip de Souza. And the untouchables said, look, our religion has done nothing for us. We are rejects, we are marginalized. But we read in your scriptures that humanity is said to bear God's image. That is good news for us. That is radically new. Because if humanity bears God's image, that means we are not untouchable. We are not inferior. We are the same. We are as valuable as anybody else. You see, this image of God concept is not some vague, theoretical, abstract, theological idea, but it is the very guarantee of the equality and the dignity of all human beings. You can have all the laws of parliament you like, but unless society has this deep understanding that upon us is conferred the likeness of God which makes us radically equal, then we will never have a just society. So that's the importance of the image of God, and we'll come back to it at the very end. But now I want to talk about some human genetics. Starting off, as I say, as a cancer biologist, and then moving surprisingly into evolutionary genetics. And then we'll think a little bit about what we can learn about some steps in the formation of our brain through history. And the story starts with a class of viruses called retroviruses. When I was a young cancer researcher, retroviruses were really making the headlines because they were being used to tell the scientific community about genes which caused cancer. I won't go into that now, but they were showing up in two other fascinating, morbidly fascinating areas. One class of retroviruses was discovered which caused a human leukemia. Not in British countries, but in certain parts of the world, southwest Japan, southern United States, parts of Africa, the Caribbean. Human T-cell leukemia virus was found to be a human cancer-causing virus. That was a fascinating finding. And about the same time, a brand new disease came to light, a terrifying disease, which filled the pages of Nature and Science week after week. And that was HIV, AIDS, acquired immunodeficiency syndrome. And within a short time, intensive study showed that that was caused by another retrovirus. So here we have some astonishing findings in the 80s associated with this class of retrovirus. So you can believe that along with just about every other cell biologist, we'd read these things week after week as the findings came out. Retroviruses have a very cunning modus operandi. That little circle at the top represents one of them in an infectious form. And the green squiggle represents its little tiny genome. About 15,000 of those A's, T's, G's, and C's. So just a little tiny genome. 15,000 is very small. And then the virus fuses with the cell membrane. That's this line which goes across the screen. And as it fuses, it's able to inject its little genome into the cytoplasm of the host cell. whether it's age or HTLV1, that's a lymphocyte, a blood cell. And then the little tiny bit of virus genome gets into the nucleus and it snips itself, it splices itself into the DNA, that's what the blue line says, of the host cell. So the virus puts its own little genome into an infected cell and then splices it into the cell's chromosomal DNA. So what that means is that every time this cell divides and passes its DNA onto its daughter cells, the virus gets passed on with it. It effectively becomes a gene. And here's just an example. It's from a recent paper where somebody has been just following this up. This blue and green line, red and blue line just represents DNA. And the TCTGTT represents 6 of those chemical letters. In this case, it's the target site which the retrovirus recognizes when it introduces its own DNA. So that thing with 2 bumps at the end represents the human T-cell leukemia virus type 1 genome of, in this case, about 10,000 bases. So the virus has its own bit of DNA up there. It selects a target site down here, and it duplicates the target site, so you have 2 target sites now, a target site duplication, and the virus DNA gets spliced in between the duplicated target sites. Can you see what happens? Selects a target site pretty much at random, effectively duplicates it, and the virus genome just neatly gets slipped inside, so that now the HTLV1 virus becomes part of the genome of the infected cell forever and ever. With HTLV1, that causes leukemia, and with HIV, that causes AIDS. And here's just an example. Oh no, this is just the strategy. So here you have a range of lymphocytes. Whoops! These cells, this represents perhaps different lymphocytes with 3 chromosomes, just for argument's sake. And the little green bar is the virus. Oh dear, sorry. Up here. And you can see that when cells are infected, the virus DNA goes most anywhere. It's pretty random. So these little green bars are all over the place. But if after many years a leukemia develops, you see that all the leukemic cells have their little bit of virus DNA in exactly the same place of the same chromosome. Now, why is that? Because of all the leukemic cells have inherited the particular bit of virus DNA from one original cell, which in this case would be that one, right? In other words, and this is known for cancers in general, so I'm not going to go into cancer biology, but time and again with human cancers, you find that cancer cells all share one or a few mutations. And they must be mutations which all of those cells, maybe 10,000 million or something, inherited from one original cancer cell. That's genetics. So here is an example. And we're here comparing a case where some people have inherited a retrovirus, because some retroviruses go into germ cells and then are passed on to future generations. So we're getting away now from HTLV1 and HIV. These are called endogenous retroviruses. We inherit them, and you and I all inherit retroviruses in our DNA, which are separate from the disease-causing ones. So here we've got human DNA. Some of us have the uninterrupted target site, and some of us have the duplicated target site with the retroviral DNA spliced into the middle. Now, fascinatingly, Denisovan man is a human creature who lived maybe 50,000 years ago. And they've been able to extract bits of DNA from this ancient human being, and they found that Denisovan man has the same retrovirus stuck between the same target site duplication in the same bit of DNA. The flanking DNA is just the same, and it goes on for zillions of base pairs, and it's just the same. So what can you conclude? If you inherit a retrovirus that somebody else also inherits, you must be descended from the same person where that retrovirus was initially added into the genome. So I can't quite remember how many of us have this retrovirus. Say it's 50-50, but for those of us who have it, whoops, sorry, I keep on pressing the wrong button. For those of us who have it, we've inherited it along with Denisovan man 50,000 years ago from the same ancestor. So that really links the human family very tightly together, doesn't it? In tens of thousands of years of history, some of us have a retrovirus, which archaic humans also possess in their DNA. And those of us who do are descended from the same ancestor as Denisovan man. That's what the power of this new genetics is. And you've seen I have got another example here. Again, some of us have the DNA with an uninterrupted target site, and some of us have the inserted retrovirus between target site duplications, and it's also there in Denisovan man and the Neanderthal man. So those of us who have the insert received it from the same ancestor as the Neanderthal. That's the power of genetics. And so we can summarize it this way, now thinking not in terms of somatic cells like lymphocytes, but reproductive cells which pass on inserts to future generations. So just imagine this as a cell giving rise to an egg or a sperm, and along comes a retrovirus, little green RNA or DNA, and that gets spliced at random also all over the genome, and then 100 years later, 1,000 years later, 50,000 years later, you find a population of people who have the same retrovirus at the same site. How do multiple people have the same retrovirus at the same site? Because they inherited it from one ancestor. That's just what genetics is. So some of us, demonstrably, are cousins of Neanderthal man. Okay, that's a fact. You can't get away from it. There's no other alternative available to genetics. Well, when did we get these retroviruses? When did they come? Well, we obviously got them from our parents. And in fact, just to do a little demonstration, if every cell of our body has 2 meters of DNA, 8% of our DNA is retroviral in origin. So 8% is 16 centimeters. So every one of us is 1/12 retrovirus, regardless of our, the color of our skin or our ethnicity. We all have inherited. This is one of the new findings of the new genetics, that we as humans have 8%, 1/12 of our DNA as being little retrovirus inserts. Now, here's something really extraordinary. We can look in our DNA and compare our DNA with the DNA of other primate species. So you see, this is where I've gone from medically important retroviruses, same principle, and we are now talking about which species share, if any, share retroviruses with us. And I'm going to illustrate this with a conundrum that has faced evolutionary biologists for a long time. In primates, and we are primates, there are anthropoids, such as us, monkeys and apes and humans. There are lemurs, which are the little furry creatures in Madagascar, prosimians, and tarsiers. And evolutionary biologists have argued like mad, what was the order of branching? Was it anthropoids branched out first as primates evolved, leaving lemurs or tarsiers, or did lemurs branch out first, the prosimians, or did the tarsiers branch out first? Well, people said, let's look to see if they share retroviruses. And maybe we can find tarsiers and lemurs possessing retroviruses which anthropoids don't have, then we would know who comes from a common ancestor. And there is an extraordinary result from a paper which came out. I just got the reference into my reference list, but I couldn't discuss it in my book. So here we have a bit of DNA, and I've highlighted the target site. And it's shared by bush babies and mouse lemurs, which are prosimians, and tree shrews, which isn't even a primate. With a number of other species, here is a retrovirus in green. And in the tarsier, it's TGAGAGAGTT, blah, blah. And it's sandwiched between target site duplications. And the tarsier has the retrovirus, that particular retrovirus, between pretty much the same target site duplications. You've got a few changes now after thousands of generations. The tarsier shares it with the macaque, the baboon, the orang, the chimp, and the human. So this retrovirus, called an MSTB1 element, is present in our DNA, chimp DNA, orang-utan DNA, baboon and macaque DNA, they are old world monkeys, and the tarsier. How do we account for a single retrovirus being shared by all these species, being present in their genome? How did it get there? It went in a single unique event in an ancestor, in fact, in a single reproductive cell, in an ancestor which is common to all of these species, but not to these ones. So the new genetics has provided us with extraordinary precise demonstration of human evolution, if you like, macroevolution, where we and tarsiers and all the species in between are descended from a single common ancestor in which that insert event from a retrovirus occurred. And here's just another example, and I won't dwell on it, but it also shows that humans and tarsiers and other species share that retrovirus, whereas bush babies and mouse lemurs don't. Even in a horse, which is a very distant relative, I might eat like one, ask my dear hosts, you can still see the same equivalent length of DNA. It's getting a bit tatty, but this again demonstrates those species which share a common ancestor. So now we can say, what's the right order of branching? Well, the lemurs branched out first. And we know that anthropoids and tarsiers share a common ancestor because the common ancestor here is the one in which those two retroviruses took up residence, because we and tarsiers share them. So extraordinary demonstration of common ancestry. And as a cell biologist reading this stuff, I thought, look, I just have to write this up and tell my fellow Christians to forget about that, not to worry about evolution, because it's established now with a clarity, a specificity that Darwin could never, never, never have believed. So there we are. There's a macaque, a Japanese macaque, there's a tarsier, and there's a lemur. And we know that we are more closely related as anthropoids to tarsiers than we are to lemurs. But some are old. Look, here's just one example. We haven't got target site duplications here because this is so very, very, very old, and it's obviously getting very tatty. But here is an element. It's called L. This boundary here is quite sharp, and you can see that this element is shared by humans, blah, 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 aardvarks, armadillos, manatees. In other words, it's an element which has been in our DNA and the DNA of our ancestors so long, it's shared by all the eutherian mammals, all mammals which have a decent placenta. So here is an extraordinary demonstration. It's getting a bit tatty, as you'd expect, because of the passage of colossal amounts of time. But it demonstrates human and armadillo common ancestry. And now people can just do number crunching. So many species DNA has been sequenced. It's all there in big databases. If you're a computer buff who wants to do your research from home at night, you can just spend your time scrolling through databases, looking out retroviruses and finding which species have them. And this is a recent study, Gemmell et al. 2015, and he's found a couple, which are shared by humans, chimps, gorillas, orangs, and macaques, 70 which are shared by humans, chimps, gorillas, and orangs, 77 which are shared by humans, chimps, and gorillas. That's the meaning of the little green box here. These are the species which have it, and therefore this is a time when it arose. And 187 which are shared by humans and chimps. He had a particularly stringent criteria which he was setting in his computer searches. And that's another example from the same guy looking at a number of different retroviruses. And I won't go into it. This is my final comment about retroviruses. I'm going to run out of time, so I'll have to miss a few slides out. Here is a very abbreviated mammalian family tree. And at this point here, a particular retrovirus was inserted into the mammalian gene line, because we've got it in the great apes and bats and carnivores and not marsupials. It was inserted after marsupials and platypuses branched off. And it gave rise to a gene which became functional in mammals. So it was a viral gene inserted into mammalian DNA, and then it was recruited to perform vital functions for at least some mammals. In fact, interestingly, in some mammals, the gene became inactivated. That's the big red cross. So old world monkeys and us, and we, the gene is still active, but new world monkeys get by without it. In mice, it's still active, but not in mole rats. In megabats, it's dead, still active in microbats. So different species can take some raw material provided by a retrovirus, and some can get by without it, others recruit it to provide vital functionality. And studies have shown that this particular retrovirus, which has a gene called GAG, and that is now known as the SIRH11 gene, seems to be involved in some aspects of brain development. So it seems, the exact mechanism is not known, it seems that, at least in us, a retroviral gene inserted at random has been recruited to contribute to the development of our brain. So junk becomes riches. This is a real rag to riches story. So people used to say, oh, these retroviruses are just junk. At least some of them are vital now to our development. And I'm going to emphasize genes a little bit tonight, simply because if our mental capacities have arisen, at least in part, from ancient retroviral infections, and our brain is the most human part of us, in a way, then how can we claim that we are made in God's image? Interesting question, I think. I'll tell you this. No, I won't. I think I better pass on. But you can see the same sort of pattern occurring with other types of parasitic agents which just copy and paste around our DNA. So there's an ALU element. It's a parasitic element which just happens to live in our DNA. Our DNA is really quite an ecosystem of jumping genes. And here's a parasitic element called an ALU element, and it uses the same strategies as a retrovirus. And you can see that this one is shared by the great apes and the lesser ape, the gibbon, but it's not there in old world monkeys, new world monkeys, or prosimians. So here is another whole set of lines of evidence for our common evolution. So we'll pass on. We needn't spend too much time with this. Now, let's look at a very ancient jumping gene. So these copy and paste. They don't come to us from outside. They just arise within our DNA, and they copy and paste. That's just what they do, randomly, but at a low level all the time. And they're building up in our DNA with the passage of time. In fact, ALU elements, what I showed you last time, we've got over 1 million of those in each set of our genome. 11% of our genome by mass are these jumping genes. So that's another sort of parasitic element. But now I'm going back a long way to these so-called amniote sign 1 jumping genes. And they were shown to multiply in our genome, particularly in an ancestor of eutherian mammals, placental mammals, in an ancestor of therian mammals, which includes us and the Aussie possums, and an ancestor of all mammals, which includes us and the platypuses. So these are really ancient jumping genes now. And these 3 boxes, these are numbers of these sign 1 elements which have been identified in our genomes. So 311 seem to be shared by eutherians, 195 shared by us and marsupials, and 100 shared by us and monotremes. And the fascinating thing is that, again, a number of these ancient jumping genes, which go in as, join the genome as junk, have now been recruited to provide vital functionality, which is involved in the development of the mammalian genome, the mammalian brain, sorry. And this might look complicated, so we'll just work through it. This represents a bit of our genome, sorry. So the black line just represents a stretch of DNA. And the boxes, there aren't boxes in our DNA, but these are just diagrams which cell biologists draw, represent special features on that DNA. So this box represents a gene called fibroblast growth factor 8. And this bigger box represents a regulatory element of DNA. So DNA contains bits of small lengths which turn genes on and off. So there's the gene down here, and here, not to scale, is an enhancer. And part of this enhancer in our DNA was derived from this amnion sign 1 jumping gene. And in fact, you can see that DNA sequences above it. So up here is the actual sequence reconstructed for this jumping gene. That's the jumping gene sequence. Below it, for all of these mammals, you can see the actual sequence now, hundreds of millions of years later, or 200 millions of years later after the event, where the jumping genes sequences have now been co-opted, transformed into vital functions of an enhancer, which in fact turns on the fibroblast growth factor gene. And that gene, this has been shown in mouse, this represents a little mouse at embryonic day 10.5. That enhancer turns on the fibroblast growth factor gene, that's the enhancer derived from the jumping gene, turns on FGF8 in these parts of the brain, and I don't know too much about brain structure, but the lateral diencephalon and the hypothalamus and the ventral diencephalon, early developing parts of the brain. So the mammalian brain, that's mouse, but it's also there in us, the mammalian brain development is orchestrated by an enhancer element, which is at least partially derived from a jumping gene which arose in a creature which was ancestral to all mammals. So again, that makes you feel a bit spooky. My brain, my human brain, is somehow dependent on a random event of a jumping gene far away in the past. But because we and platypuses share a remnant of that jumping gene, we know it occurred in a common ancestor. And another story driving another protein which drives another event, this time in a day 13 mouse embryo in the neocortex. So another evidence of one of these jumping genes contributing towards regulation of mammalian brains. Now, 20 past 8. I think I'll just flick through some of these. I'll finish with this final story. That's a remarkable diagram. I've unloaded it from the National Institutes of Health website, where they have an image gallery. And they're very generous in letting you use their images for educational purposes, so long as you acknowledge it. And that is a very recent picture. It was published in Nature magazine, in fact, about a month ago, showing domains of the human brain. And touch is this part of the human brain here. Hearing is part of the human brain here. And sight at the back is involved there. So using brand new, very sophisticated imaging techniques, people can see what parts of the brain are involved in particular sensory functions. And here's a relatively recent paper or study which indicates how parts of the brain have arisen. And these are genetic changes only in human beings. So here's part of chromosome 1. That's chromosome 1 here, this big box. And there's a gene called SIRGAP2A, which is located just about here. And only on the human line, because it's not present in chimpanzees, this gene, or part of this gene, that represents the gene, that box, part of the gene, represented by the blue dashes, was duplicated to form a daughter gene. This sort of thing happens. It's well known in genetics. And then that was in turn duplicated, like so, to form a third gene called SIRGAP2C. So genes can be duplicated also in genomes. And it's been found that SIRGAP2C interacts with SIRGAP2A to cause subtle changes in the development of brain cells. Now remember, this is in humans only. So this may be contributing towards the development of human-specific features of the brain. So that's a 2012 paper. And this is my little cartoon trying to just simplify the paper. Our nerve cells have long processes, long threads coming out of them by which they communicate. And on those threads are little structures called spines, so that the nerve cells in our brains can talk to each other. There are little knobs called heads on little arms called necks. And SIRGAP2A is believed, this is the original gene, which we share with all other mammals, is involved in the formation of heads and necks. SIRGAP2C, which is the duplicated gene, has subtle effects. Initially, when it's active in nerve cells, it shrinks the heads and increases the density of and the length of necks. Why should it shrink the heads? A big head is good for nerve conduction. Well, eventually, it's been found that the presence of this duplicated gene, duplicated only on human beings, leads to delayed formation of heads. So it promotes the development of nerve cells with all of these little spines so that nerve cells can communicate with each other, but it delays it. Now just think how long human beings are dependent on their parents. So this could be a human-specific effect, enabling a longer childhood so that children can learn from their parents, which is a particularly and uniquely human facility. So we have here a human-specific genetic change, that's a chemical change in our DNA, with implications for our human-specific ways of learning and knowing. So that's all I want to say about genetic. As you can see, I've had a little bit more than I really had time for. And I'll just leave the genetics with this point, that human beings show this feature called neoteny, which means delayed development of our children, of us too when we were children, allowing time for learning and socialization. And so we leave genetics with the conclusion that genetics here seems to be giving away to relationship. So the genetics is as rigorous as it can be. It tells us that we are descended from a common ancestor with other apes, other primates, other eutherians, and with all other mammals. The genetics shows us that. It's also showing us steps in the formation of the human brain. So the next question is, well, where then is my human dignity? I mean, am I different from another ape? Because my brain has arisen the same way as an ape's brain has arisen. Well, first of all, here's words of comfort from a recent book by an American theologian called Philip Rolnick. He says, rightly approached, science will become a benefactor. I would say science is a benefactor to the Christian faith of our time. A careful contemplation of scientific discovery can bring us to a broader and deeper love of creation, and I would say of the Creator. And so here's another picture of the brain in its metabolic activity, functional MRI from the National Institutes of Health, and we raise the question, are we just apes? In fact, as sometimes you might have heard someone say, am I just a glorified ape? Are you just telling me I'm a glorified ape? Well, let's move on. Here is from a very recent book. Anybody read this book yet? It's a beautiful book by Wagner and Briggs, one of Britain's top artists, top religious artists, and the professor of nanotechnology at Oxford, Andrew Briggs, a brilliant scientist. And they've written this book, 2016, The Penultimate Curiosity, How Science Swims in the Slipstream of Ultimate Questions. So that's a wonderful book. And they quote Sir Henry Ackland, who says, whatever views or hypotheses or guesses you may have about the origin of human beings, you are all agreed that however he so became, he is in some manner made in the image of God, a spiritual being. So I agree with that. So if you're worrying about where is room for humanity amongst all this genetic stuff, all this mechanistic stuff, just hang on to Sir Henry Ackland for the moment. You needn't be anxious. Because when I started to think about neoteny, it brought us to a whole new subject area. Genetics takes us so far. Yes, genetics produces genes which make proteins, which interact, which create the fundamental substrate of a human brain. But they leave room for human brains to develop in response to personal input, to social learning, to a mother's love. And here from a very elegant little paper from 2015, The Social Life of Cognition, interactive communication, that's the things which we human beings do specifically, chimps and gorillas and aardvarks don't, is responsible for the development of language, of theory of mind. Theory of mind is our ability to see behind the face we're looking at and to recognize that there is a mind as we talk to each other, just like mine, and so to understand your perspectives and your thoughts. Theory of mind, people, it may be there in a rudimentary form in other creatures like dolphins or chimps or whatever, but theory of mind is supremely developed in human beings. That requires interactive communication. Genetics is not enough. The mental representations of time, motion, space, musical pitch and colour, these are learnt by exposure. They are not genetically encoded. And our ability to attend, to learn and to memorise. These things are not genetically encoded. They rather rely on interactive communication. So genetics takes us so far, they leave us poised to develop social brains in response to social inputs. And this is a lovely little paper from 2014, again in Science. That's my words up at the top. Genes are necessary but not sufficient for the development of a human person. These top psychologists say most, possibly all, human neurocognitive skills, our ability to think and relate, are shaped by culture. And many are culturally inherited, but the parallels between mind reading and print reading are extraordinary. So these people say to become fully human and to develop theory of mind or to be mentalized is very much like learning to read a book. Our genes don't teach us to read books. Not even our human-specific genes teach us to read books. We read books when we are taught. And when we are taught, the appropriate neuronal connections are formed in our heads, which enable us to make sense of those little squiggles on sheets of cellulose. So these clever scholars are saying our ability to learn to read a book requires input, personal input, from those who are human before us, just like our ability to relate to each other as persons. It's not genetically encoded, but it's socially transmitted. It's the function of relationality. And they just give, this is a partial table which they produce. I've just taken some of the more obvious parts, where print reading and mind reading, reading a book and reading minds, show parallels. They show different neural areas. Disorders are there, like dyslexia is in the case of print reading, or autism in the case of mind reading. They continue to develop to late adolescence. They require cognitive effort. We don't sit down and, we don't work on our kidney developing or our liver, but we do with our brain. We have to sit down and learn how to read and practice it, just as we need to practice engaging in relating to other people. There are, of course, individual differences, but these arise from the environment rather from genetics, unless you have a very serious mutation. And of course, there are rules. So they say, fascinating stuff. If a group of human infants managed to survive on a desert island in a cruel Lord of the Flies-like experiment, they would be no more likely to develop a theory of mind and become explicit mind readers than to develop a writing system and become literate print readers. So whereas the primordia of our brains arise through genetic events which evolutionary geneticists can determine in very fine genetic detail, that doesn't make us human. Our genetics is extraordinary and enlightening and wonderful, and we should take it all very seriously. But it's not the whole story regarding our humanity. It doesn't take us to being the image of God. Now again, I'll have to hurry because I've got more material than I've got time to give. But we can see these tragic experiments where human beings are deprived of social input. And when these experiments are studied, people find that a child brought up neglectfully, without personal experience from the generation before, grows up severely neurologically malformed. So we have this extraordinary, and I think as a Christian, this incredibly insightful understanding that the very structure of our brains is formed by spiritual and social relational qualities. And there's a 13-year-old boy, that's a public image domain from the internet, brought up in a Romanian orphanage without the intense love and compassion and communication from a mother. And his brain, his behavior, his language, and his brain have not developed. Here's just one study. In fact, I think this arises from the University of Southampton. So anybody from the local psychology department here? Just stand up and correct me if I'm wrong. But here they've divided children into severely deprived, more than 6 months in an orphanage, and less deprived, less than 6 months in an orphanage. And they have studied them for ADHD, attention deficit hyperactivity. And they find that low deprivation children have 5.6% of them suffer from ADHD, which is pretty much the same as the general population apparently. High deprivation children, 3 times as many at the age of 15 show ADHD. Does this disappear as they continue to grow? The experiment has now run out to 20 to 25 years. Low deprivation children, about the same, 3.8, even a bit lower. High deprivation children, at least as high. This is a permanent neurological change as a result of deprivation in a small child. Just reminding us the inadequacy of genetics to make us human and the requirement for the input of parents, communities, teachers, love. And so, I mean, that's just a graph which depicts the same thing. And now I'll just flip through the next few slides. So we have this powerful association. This is the summary of this paper. A powerful association of early life experience and later development in a way that suggests a role for deep-seated alterations to brain structure and function. That's mind-forming matter. Similarly, here's a group where they've looked at IQ and theory of mind, and IQ and theory of mind are both more poorly developed in children deprived of social input. DNA methylation, we'll pass on from that. That's another NIH picture showing how they can look at tracts in the human brain, white matter tracts, and how a white matter development is abnormal in children who are neglected. And so are grey matter tracts, abnormal. That's grey matter is cell bodies in the brain, abnormal in children who are neglected. And you find particular regions of the brain. The hippocampus is shrunken, the amygdala is increased, and there are also abnormalities in these tracts, the anterior cingulate cortex and the caudate nuclei. These repeatable now changes in the brain structure of kids who are deprived of social interaction. And head circumference. So that brings me to the end of my talk, and the question is, well, these are still children who've, been exposed to human contact of some sort. What happens if you really deprive them of input? And that's known as the forbidden experiment for obvious reasons. To take children and to deprive them of social input would be a highly unethical thing, but it appears that Frederick II of Hohenstaufen did that about 800 years ago. He wanted to know what language children would speak if they were brought up in total silence. And we have the record from Friar Salambini, who has told us the results of that experiment. And as far as we can understand from Salambini's report, children who are bathed and washed but who are never spoken to simply did not flourish, and they died. This is Salambini's words, and you can find it in a number of histories. Frederick bade foster mothers and nurses to suckle and bathe and wash the children, but in no ways to prattle or speak with them, for he would have learnt whether they would speak the Hebrew language, which had been the first, he thought, or Greek or Latin or Arabic, or perchance the tongue of their parents of whom they had been born. But he laboured in vain, for the children could not live without clappings of the hands and gestures and gladness of countenance and blandishments. There you go, another experiment down the tubes. very familiar to us scientists. But tragically, it seems that children died in the absence of communication. I did a theology degree with the University of South Africa. Never been to South Africa. But I was told that for somebody who was overwhelmed with work, there was a very good university that sent your letters to you. This is before the internet was thoroughly developed. I finished in 2000. And I enjoyed my BTH. And one of the most important things I learned was this African saying, which has such wisdom. I think that machine-minded scientists sometimes need to stand back and listen to folk wisdom. Umuntu ngumuntu ngabantu apparently means a person becomes a person through persons. I think there's such incredible wisdom in that saying. You hear molecular fundamentalists speaking as if genes control everything we are and do. You know, everything, love and attraction and all that sort of stuff is encoded in the genes. That's all we are, just a network of genes. Rubbish. This is such wisdom. A person becomes a person through persons. And that's where I can accept everything that genetics tells me, no matter how mechanistic it is, and wonder at it, but knowing it still does not tell me how or why I can express the fullness of the image of God as a person who communicates, who can learn to love and show compassion or whatever. Which brings me to the words of Jesus. I find this so compatible with the words of Jesus. We become human not by in genetic inheritance, but by knowing. And in his high priestly prayer before he was crucified, Jesus prayed to his Father on behalf of his disciples. And Jesus said, eternal life, this life of the new age, this different dimension of life which Jesus came to offer, means knowing you, the only true God, and knowing Jesus Christ, whom you sent. And when I was younger, I used to wonder, life is something tangible. I'm a living thing. Bump, bump, you can feel me. There's a heartbeat there. And yet knowing is so ephemeral. What is knowing? How do you know somebody? But from what we've been talking about, we know that knowing is what forms us as persons. And Jesus, speaking to his Father in heaven, was saying that to know God can be known. That was something that Jesus emphasized. To know God is to be formed as a person in a new and deeper and more wonderful sense. And that's why we can rejoice in genetics, but we can look beyond it to the power of relationality as that which forms us completely as human persons. Thank you.